Hello, good morning, good afternoon, good evening, and good night. My name is Andrew Moyer with AskingPennState. Welcome to the first episode of our second season. Have you ever thought that the blood from certain animals or creatures could help forensic sciences and solve murders? In this week's episode, we discover how the blood from leeches can be beneficial to us. More specifically, how they can be advantageous to forensic sciences. The research done by Elizabeth Knapp, a third year here at University Park, goes into more detail on this. So stay tuned and enjoy the video.
My name is Elizabeth Knapp, and I am a biology major at Penn State University in the Schreier Honors College. And what got me into my research was that I stumbled upon a case in Tasmania, Australia, where a man named Peter Cannon robbed an elderly woman. And as he was escaping, a leech got caught onto his leg and engorged itself on his blood, and then fell off, and then was later collected by the crime scene investigators. And when he committed another crime years later, they kept the leech on file and used his DNA that they received after he committed the other crime, matched them, and it was a perfect match. So this was one of the first instances in which a leech was used to identify a person. One of the coolest things that I found about the research was the fact that we had to first determine that the DNA DNA inside the leech's midgut was in fact human and was in fact not leech blood, but human blood. And I think that was my favorite part about this research, was that we had 3 tests. The first was called leukomalachite green, in which it is a presumptive test where you use a Q-tip and you use leukomalachite green solution. And if the Q-tip turns green with the addition of blood, that means it is blood. With the addition of a substance, it is blood. And then following that, we had a confirmatory test known as Takayama, which is so cool. Under a microscope, if it's blood, it has these beautiful crystal-like structures. And it's called ferroporphyrin, the crystalline structure. And so that is a confirmatory test that it's definitely blood. And then we use this final species-specific test called hexagon OBTI. I connected in my brain like a pregnancy test, in which there's a control and a test line, a C and a T, in which you drop the blood on the control, and if it moves up through the T to the T through pulling the substance up, it will make 2 lines to confirm that it is in fact human blood. But what's interesting is that ferret blood will also make 2 lines in the assay. Isn't that so interesting? Because I guess we have genetic similarities down to a very, very small level to ferrets. And so it will also produce a positive result. I think there's so many pros. If I could pick one pro to researching, it would be just being able to learn. I think for as long as I can remember, I've always loved learning. I love school. I love learning about different things. And I had, for the Schreier Honors College, we are expected as biology majors, as majors of science, to get research for our senior thesis. And when I stumbled upon Dr. Roy's resume on the Department of Biochemistry and Molecular Biology website, I was immediately interested, and she got back to me right away. And we set up an appointment, and we hit it off from the start. I'm so blessed for the opportunity. So there's so many pros, but I think the biggest one is learning and being able to learn every single day. And the way that she's so patient when I make mistakes, I'm so grateful, because I make them a lot, but always learning. When it's 0 hours post-feed, the leeches are put into the freezer immediately after we put them in the vial of the 0.5 milliliters of human blood from one male donor. So they're immediately euthanized, immediately put in the freezer. However, with the 24-hour post-feed euthanization, they're allowed to feed for 24 hours, in which we're also unsure how the enzymes are breaking down the blood within their stomachs, within their midguts, within their crop area. We're unsure. That's another thing we're unable to quantify. So this research included a lot of retests and reamplifications within the PCR system to ensure that we were able to get complete profiles at each time period, no matter if it was 0 hours post-feed or 24 hours post-feed. We use known reference samples to match them to our YSTR samples so that if they're a complete match, we can determine that it's a complete profile. But if there are certain alleles missing, then it's an incomplete profile, and we call that more informative when there are only like 2 alleles missing. But if there are more than 2 alleles, then it's a less informative partial profile. YSTR markers are revolutionary in the way that they can clearly distinguish individuals, one individual from another, in that it's DNA, but it's more than that. They're short tandem repeats in that while each person has a different set of genetic material, different set of genetic makeup, of course, except twins. Short tandem repeats, there's a different amount of them in each person's genetic material, in each person's DNA. And so they're so important for forensic researchers, for forensic analysis, especially because of why, because it can be used for males and it can be distinguished between female and male. So you can of course do regular STR without Y, but YSTR is crucial for our research because we used one singular male donor. Honestly, I could only say good things about this research, and I owe it all to Dr. Roy, honestly, my PI, and the other members of the lab, my thesis supervisor, Dr. Michael Axtell, who is a member of the, he will be my thesis, he will read my thesis for the Schreier Honors College and will hopefully approve it someday. And Veronica Capez, who is a graduate student in the forensic program at Penn State. She has been so instrumental in our research as well. We presented at the Northeastern Association of Forensic Scientists Conference in Rhode Island, and Veronica was the second author on our research, along with Dr. Roy, who was the PI and overall supervisor. And so I'm just so grateful to have their support and to have their confidence in me throughout this whole journey, because this has been my first time in a lab ever. So to have it go so amazingly and to learn so much in such a short amount of time is just awesome in my eyes.
Thank you so much for watching this week's video of Asking Penn State. We will now be posting bi-weekly on Sundays. Our next video will be published on October 23rd, and it's a fun one, so keep an eye out for that. Signing off, I'm Andrew Moyer. I hope you have a great morning, afternoon, evening, and night. Thank you, and I'll see you next time.